decent amount of folks have shown up, so we can probably get going. So I'm very excited today to introduce our speaker, Lorin Crawford, who's a senior researcher at Microsoft Research and an assistant professor in the Department of Biostatistics at Brown. Take it away.
Awesome. Thanks so much for having me. It's my second time, so I missed me. I didn't do too bad of a job the first time I gave a talk in the department, hopefully. Hi, everyone. It's great to be here, even virtually. Today, I'm going to talk about another side of my research program. So when I first came, I talked a little bit about non-parametrics and variable selection type stuff. And today, I want to talk a little bit about another passion of mine, which is building statistical frameworks for this idea of mapping three-dimensional shape variation onto genotypic or phenotypic variation. And so for those who may be unaware of what I do, a recurring research theme in my research group and in my research program is to take modern computational approaches and develop theory that enable their interpretations to be related back to principles. Like many people in this department, I don't just think about biology as a tool and we used to use my really fancy hammers. I really think about building specific hammers for these given nails, right? And so classically, I think of this idea of dissecting phenotypic variation. And what I typically what I would maybe consider like a little bread and butter of what I do is, think about this in the context of complex traits for human beings and also model organisms. So, if we have some phenotypic variation, you could dissect that variation of pi into things like additive effects, understanding like pairwise interactions, gene A times gene B effects, third order interactions, and maybe how our genetics play a role in terms of interacting with our environments to give rise to complex traits. So I like to show this plot here because these are phenotypes from different mice over these different categories. You can see that kind of, if I break down these genetic architectures, you can see the variation across what types of genetic effects play a role in understanding, it's playing a driving force in that variation across these different populations. Well, today we're going to talk about something very similar, but not just with like continuous quantitative traits, but with shapes. And so you can think about the same kind of idea of modeling variation, but now our phenotypes or the things that we're going to look at are morphological. Okay, and so on the left-hand side we have these bird beaks from different finches. On the right-hand side, we have heel bones from different primates. And so the idea here is that we want to build statistical models that allow us to understand and do almost like 3D variable selection on these like surfaces, these three-dimensional objects, and understand variations across different groups of whether that be primates, birds, or the case might be. And so my presentation outline is going to be broken down into two parts. Okay, so first I'm going to walk everyone through this idea of previous work with shapes and statistics. So we'll talk about the history of comparing shapes, this idea of topological summary statistics, and I'll walk you through this idea of how we use TDA. I'll go through a prediction-driven application in radiomics. And then we'll talk about this pipeline that we created called SINATRA, and I'll get a little bit about the things past just the name that I kind of love, which is a pipeline for doing variable selection on 3D surfaces. Okay. We'll talk about the algorithmic overview of this. I'll bring back or revisit this idea of the relative centrality measure. Some of you might remember this Michael Jordan slide. We'll get back into that just so people can re-see this example. And then we'll talk about how we take 3D objects of different classes. What we want to do is identify almost structural fingerprints of these shapes that are describing the variation between these two groups as a way to do variable selection. So we'll talk about how we take these shapes, turn it into topological summary statistics, do variable selection on those summary statistics, and then reconstruct the shapes to kind of see visual enrichment of structural fingerprints of these different phenotypes. And then I'll show you how this works with simulations and real data. Okay. So yeah, let's have some fun. So starting with previous work with shape statistics. So the history of shape stats is actually pretty interesting. So classical shape statistics used to take these like 3D surfaces, and what you would do is you would place landmarks, almost if they were expert derived landmarks on these manifolds, and you would say that I have a lower dimensional representation of this shape based on these placed landmarks, right? So on the left-hand side, we have a skull. Each of these points here is a landmark that kind of describes almost like a significant marker or feature of this skull. And then what you could do is you could take the landmarks as like a lower dimensional representation of what you see on the left-hand side. right? Now, obviously, user-defined landmarks create a whole ton of problems because you have a slew of heterogeneity that can happen, right? If I have two different experts, I might see two different placements of these landmarks. Also, if I show the same expert the same skull again, I might have a totally other set of landmarks also that they place, right? So we moved past user-defined landmarks and moved now to like these semi-automatically defined landmarks. And that bypasses this variability issue I just raised, right? This is typically used in this idea of biological morphometrics. So on the bottom here, we have teeth from different primates, a carnivore and a herbivore. What someone has done who studies these primates, what you can do is you can put, it's like almost like a diffeomorphism between these two shapes. You could do and say, I have a correspondence where I know that this particular feature is different for this species versus this species, to do that across the shape. And what you can then do is define a distance function, right? Now that I have a correspondence map between my two shapes, I can define a distance function, say Percusses or something that says how close in some space are these two shapes to each other, right? So these automatic landmarks are cool because it's giving us a way to kind of compare objects, right? And there's an issue with landmarks in that what I want to do is think about if I have shapes with a given phenotype that's attached to those shapes, right? Like almost like maybe a survival metric, if I think about radiomics, or I have some feature that I've studied about these primates. So what I really want to do is take all these shapes together and think about how to have a representation for them that allows me to do variable selection with. Well, landmarks are hard to work with, right? Because they're these triplets, right? And the idea here is like, how do I take an XYZ coordinate and plug into a regression model where I can have an effect size for which I can do variable selection with this? It's hard to think about the geometry of that space, right? And so what my group takes advantage of is this idea of shape representations, current shape representations, where we're allowed to represent three-dimensional objects now as meshes. And you're gonna hear me say this a lot, this idea of a collection of vertices, faces, and edges. Okay, and so here what you see in the bottom is a brain and a brain tumor, but you see we've kind of triangulated this mesh, right, into this idea of vertices, right, these individual components, edges between these vertexes, and then the faces. Okay, so my group builds methods to kind of summarize data over these kind of representations of shapes. Okay. And so there's some main objectives that I think about when I want to build methods in this context, okay? One, I want my transformation between three-dimensional object to summary statistic to be such that I can use a wide range of regression and machine learning techniques. And that stems from GLMs all the way down to neural networks, or whatever the case might be, and everything in between, right? The desired properties for my transformation, I need my transformation from shape to summary stat. One, I need in my summary statistics space to be able to build probabilistic methods. So I need to be able to compute distances, define probabilities in a transform space. But the most important is there's a first point. I need my transformation, that function from shape to summary stat, to be injective, or even better, just explicitly invertible, right? I need to be able to say, once I take a shape and represent it as a summary stat, I need that function to be able such that once I do statistics in that summary statistical space, I need to be able to transfer back into the actual surface to be able to visualize what might be happening in terms of like the actual data, right? So what we're going to do is we're going to mess with topology, okay? And we're going to work with two topological invariants. There are a ton of them out there, but we're going to focus on the bottom two here, the persistence homology transform and the Euler characteristic transform. And I'm going to show you visually how these things work in practice, and why we focus on the Euler characteristic over the persistent homology transform, okay? And I like to motivate topology with this Picasso painting, because what we're effectively doing is we're taking these very complex objects on the left-hand side, and basically working in the space of understanding holes in space, like on the right-hand side, as a way to understand these like really complex things on the left-hand side. And this Picasso painting is a very classic way of people kind of understanding and visualizing the usage of topology, okay? Now, persistent homology is this idea of studying the persistence of features, okay? And we're going to work with this over what we call a simplicial complex. And the simplicial complex is very easy. Again, going back to what I say for my mesh representation, simplicial complexes can be based on, again, vertices, edges, and faces, right? And then tetrahedrons. But again, collections of vertices, edges, and faces, okay? So what you do is you can study these simplicial complexes according to these Betty numbers, okay? And what we actually really care about is when I look at a shape, I want to understand the connected components of the shape. I want to understand the holes in the shape, and then potentially voids, okay? In persistence homology, the idea of this is very simple. Okay, so what I do is I start with a point cloud or some points on an object. And what I do is I grow balls around that object, around those for some vertex around each ball. And as I grow these balls over some filtration, I'm going to start seeing these balls connect to each other, right? And I'm going to basically, the idea of persistence homology is I'm going to track the evolution of connected components, holes, and voids across those filtration, right? And so there are many ways that you can kind of track this. The typical way that people look at this are barcodes, right? So again, I start with many different points on in my point cloud space. I grow these balls over these shapes. I track how long certain features persist over that filtration, right? And that could be how long until I get one long connected component with H0. H1 is how long do certain circles or holes in my object persist over time. And then H2 is how long do I have a particular void. Okay, and I do that until I have just one long connected component. There's another way to track this is what we call a persistence diagram. So it's basically the same idea of tracking birth and death time of these barcodes, but now on a XYZ plane, okay, or XY plane, two-dimensional plane. So what we have is, I'm going to show you how this kind of works in practice. I have a two-dimensional object here. What I'm going to do, instead of growing balls around my object, I'm going to take a filtration up through my object. What I'm going to do here is the same kind of idea. I'm going to count when I see a feature. That's going to say when a feature is born. I'm going to continue to filter up this object, and I'm going to see when certain features become connected to each other. That's when a feature dies, right? And then I'm just going to have one huge connected component at the very end. Okay, so I'm going to start my filtration at the bottom. I'm going to filter up. I see my first feature. I track that as the birth time of that feature. I continue up. I see another feature. See, I've tracked this on the x-axis. I come up. I see another feature. Now that feature is going to be short-lived, right? Because as soon as I move up, it's going to be swallowed up by the feature before it, right? So that feature now dies, right? It's become connected to the feature that I saw first. Okay. I'm going to scroll up. I see another feature. That feature is also going to be short-lived, but we're going to see one more feature first. Again, the feature I just saw is going to die because it's going to be connected to the feature before it. We're going to see another death, and then I have just one large, huge connected component. Okay. Now, the idea here, when you do these kind of filtrations, is that things that are short-lived, like things along the diagonal that are born and die quickly, are what we consider to be topological noise. There's some issues, there's some disagreements of how we should interpret this in the field, but by and large, that's how we interpret it. Things along the diagonal are things that are less interesting. These are things that don't survive for a very long time. Things far off the diagonal are things that we find interesting. They're the things that persist over some time. What we do is we focus on the off-diagonal things as being the things that are driving this topological signal. Okay. Now you might be asking, like, what can I do with this? Well, there's actually kind of a lot of cool things you can do with TDA. So it turns out, actually, this is from an internship I did in Durham as a grad student. You can study the topology of how maize roots grow as a predictor of how much yield they'll actually produce. And so if you're thinking about how to derive crosses for things, you can actually just study these trajectories of these maize roots and then say, like, I can predict how much yield this thing might. It's actually a really good predictor of this. If you're trying to understand on a topological level, certain crosses might lead to the same yield production. You could do this via topology to see how close in topological space they actually are, right? To see how similar they actually will become. So in terms of yield production. So what we do is we do same filtrations. We derive these persistence diagrams. You compare across them and see which crosses might lead to certain better yield production. And then maybe if there's two crosses that lead to the same yield production, but one is cheaper to produce, you might go with that, right? Or more robust to different environments, you might go with that. Now that's a 2D example. Now the persistence homology transform, the PHT, is the same idea, but in 3 dimensions, okay? So how this works is the same idea. I have a 3-dimensional heel bone here from a primate. What we're going to do is I want to study the topology of this shape. So what I'm going to do is the same idea. I'm going to define a filtration, a height function, and filter over the shape. What we're going to do is I'm going to filter over. I'm going to derive the persistence diagram for that shape. I'm going to collect it. I'm going to save it. I'm going to rotate the object. I'm going to do another sweep. I'm going to take that filtration, that persistence diagram for that filtration direction, save it, and do this over many directions of the unit sphere. Okay. So what we do is we define a height filtration function. I have a direction new. I'm going to sweep over. I take that diagram, I save it. I rotate the object. I do the same thing again. Take that diagram, save it. And I do this over many directions of the unit sphere. So now my persistent homology transform is the idea of having collections of persistence diagrams that describe the geometry and topology of a shape. Okay. Now in practice, what you would, in theory, what you need to derive to have the entire shape understood is you have to take infinitely the many directions over the unit sphere, right? In practice, there's a lower bound for how many you actually need to use these persistence diagrams as a sufficient statistic or summary statistic of the geometry and topology of these shapes. Now, what do? Once I have these collections for diagrams for many objects in my data set, I can just now study how similar heel bones are for different primates, right? So here we have heel bones for 67 different genera. What I could do is do these collections of diagrams, figure out the distances from them, plot these on like some two-dimensional plane, and I can overlay and see that certain species phylogenetically cluster together with each other. Okay. Now, what's my issue still with the PHT? Well, the PHT still doesn't give me kind of what I really want, right? Like it's nice. I can now compare three-dimensional objects, but I'm still kind of left with this landmark issue, which is, common regression techniques have a nice inner product structure for their covariates that are defined in some Hilbert space, right? The PHT still gives me a birth and death time pair. I have these ordered pairs, right? So it doesn't have a simple inner product structure where I can think about things like an effect size, right? So it's still hard to really work with. And also have these collections of diagrams, so that makes things even a little bit tougher, right? So what my group really focuses on and works on is the Euler characteristic. It's a different invariant with the same properties as a PHT and the persistent homology transform, but I get a single numerical number, and it's a very easy computation. What you do for every sweep in your data set is you just count the number of vertices, edges, and faces at that given time. Vertices minus edges plus faces, and I get a number, and that summarizes things. And so I can do this over many steps over an object, and I can count basically what we're going to call the Euler characteristic curve. Okay. Now, how does this work? Let's take a 2D example. Here I have a hand. I'm going to do these sweeps over this hand the same kind of way, and I'm going to create what we call an Euler characteristic curve. So I'm going to start my filtration. I'm going to filter across, and I'm going to count the vertices, edges, and faces over every single step of this filtration. What I'm going to do after I'm done is I'm going to rotate the hand, do it again. I'll get another curve. And what we'll do is every time I rotate the hand, I'll just concatenate curves together. Okay. So at the end of this, I have this long concatenated curve over all of these different directions over the unit sphere. I wish I could like swing my arm around, but I can't. See all these different directions over the unit sphere. What this does is it gives you a design matrix, right? Every row now is a concatenated curve for that given observation. So I'm thinking about P being like the number of topological summary statistics that I have. I'll have an N by P matrix, which is in the exact same structure that we're used to working with in terms of data, all right? The Euler characteristic is also invertible, right? I can also just say, like, I can understand over any given direction which topological summary statistic came in what step of the filtration and from what direction. So I can also think about how to map back onto my shapes. And so the Euler characteristic is nice because it has this nice inner product structure. And now we can also think about how to use the full scope of parametric and non-parametric methodologies. Okay. And you might really like, what can I actually use any of this stuff for? Well, let's see a simple example of an application of radiogenomics. Okay. So here we're going to have MRIs from glioblastoma patients collected from about 40 people. This is data that's publicly available in the TCIA. This data also has matched genomic and clinical information in the TCGA. Okay. So here's kind of what the data looks like. We have an MRI scan. What we do is we segment out each tumor. So we have slices of these segmented tumors. And we're going to run our topological summary statistics over these slices. And so we get these nice curves for these different individual slices for every person, right? So every person has this nice concatenated curve. What we do in our group is we think about nonlinear models, right? So I have these nice curves. My group likes to model things with GP. So we have a nonlinear model here. So not this like x beta, we have some f of x. Let's put a prior this GP over this function space. And here you can think about the covariance function of my GP as trying to summarize the similarity between the topological summary statistics of patient 1 versus patient 2. Okay, and we could just use like a Gaussian kernel to do this. Right. Now, the topic of this particular paper was trying to get at this idea of are topological summary statistics useful? And in the context of glioblastoma or radiomics, are these more predictive of things like disease-free survival or overall survival, more so than things like gene expression, tumor morphometry, volumetrics, geometrics, these kind of things that are classically taken in this space, right? Is topology a more robust predictor than these things? And so what we do is we take disease-free survival, which we say is the time from a patient was diagnosed to the time that the patient relapsed, and overall survival, which is the time that the patient was diagnosed to the time the patient was no longer with us and they passed away. We're going to do these 80-20 splits 100 times, and we're going to use predictive root mean squared error for this particular topic. Okay. So here are the results, and this is going to springboard us into where I came from with SINATRA. So this disease-free survival, as you kind of imagine, first of all, we can look at all the results. So root mean squared error is the first column for each of these colored columns. The probability optimal means that out of these 120s, or out of these 100 splits, which of these particular covariate types was the most robust, or was always the best predictor. So you can see these kind of numbers. Overall survival is murky. I mean, I would argue that there's no difference between these methods of these data types for overall survival. For disease-free survival is pretty interesting. And glioblastoma actually is a nice use case for this, where radiologists have seen that holes in a tumor can be signs of more aggressive necrosis, which is also signs of stronger things like IDH1 mutations and things like that, right? Which lead to different properties and stuff downstream. So what this kind of helped me start to think about was maybe there might be a nice connection between morphological signatures and maybe signatures that are happening on a genomic scale, right? Like prediction's nice, but maybe it'd be cool if we did like interpretability here, like interpretation would be kind of better, right? And so this got me thinking about this idea of oncogene activity and therapeutic resistance in the context of how these things might manifest themselves in terms of signatures and shape. I probably have to go about this with this group, but just in case you may not know what I'm maybe speaking about, in our cells now are like well-oiled machines, right? Where if I think about a signaling pathway with different genes, in our regular functioning cells, some growth signals come in. Gene A talks to gene B talks to C talks to D, and D is always telling the cell it's okay to proliferate and grow, right? Well, in a cancerous system model, so you have some mutation. So this pathway now is consistently active, which means that B is always on, C is always on, D is always on, survival is always proliferating, happening, and this is how you get irregular growth, right? Now, a lot of areas, a lot of cancer subtypes now, we have these targeted therapies, right? These agents that come in and block the activation of certain mechanisms, right? And so what happens in this particular cartoon here, what we're doing is we're blocking the activation of B. B's now off, C's off, D's off. Survival and proliferation is no longer happening, and this person looks like they're healed, right? Now, therapeutic resistance happens when this pathway somehow either turns back on, either by some like feedback loop, or other pathways in the cell that do similar things also get turned on, right? Or maybe something downstream is happening. Anyway, survival and proliferation is always happening, and then irrespective of this drug still being applied, this patient now, the drug now has tumor resurgence, right? And we have therapeutic resistance occurring. So this radiomic study tried to, in my mind at least, might have pointed to is that there might be a way to connect this process to like tumor shape. And so here we have a mesh from the same TCGA study, done a lot better in terms of like shape representations, where now we're actually take each of these data sets, we run them through these convolutional neural networks and actually reconstruct the tumor mesh as if I had like gone into your brain and like pulled out the tumors. So actually we have these really nice constructed meshes based on these libraries at UPenn. And so the idea here was to move past prediction and think about variable selection here on the 3D surface. Okay, so that brings us to SINATRA. SINATRA's acronym stands for Sub-Image Analysis of Topological Summary Statistics. For me, it has a little bit of a deeper meaning. For those who know me, I do a lot of things. I think about cancer and genomics. I think about statistical genetics, and I also think about non-parametric Bayesian type methods, and I also think about this TDA type stuff. And SINATRA is like the first project I think that takes all of these weird disparate sounds and brings them into like one cohesive musical project. And so this has a little bit of cool of a double meaning to me. But SINATRA is a very simplistic pipeline. It's for how we think about doing variable selection on 3D shapes. So we have some input data. You can think about these meshes. Here we have teeth again. Let's stick in this teeth space where we have teeth from two different primates, a carnivore and a herbivore. What we do is each of these collections of meshes is we turn them into topological summary statistics. So we do the same kind of Euler characteristic. We do these sweeps. We create these curves for each mesh in our data set. What we do now is we have sets of curves for the two species. What we're doing is we're going to do variable selection on these curves. We're going to identify the pieces of these curves that best describe the variation between these two groups. And again, because I said the Euler characteristic is invertible, what we could do is we can take the significant pieces of these curves and map them back onto the physical surfaces themselves. That's what you see in panel C, thus giving you D, which kind of highlights these morphological signatures of what separates a tooth from a herbivore versus a carnivore. Okay. Now, so this is the steps of SINATRA, but remember, SINATRA is just a pipeline. And I like to say this, especially when I speak to people who are methods developers, is that SINATRA is a pipeline. I'm going to present on how we fulfilled these given steps. If you hate any of these steps, fine. Take out that pipe and replace it with your favorite one, right? So SINATRA's 4 key steps are very simple. We just want to take a shape, represent that shape with summary statistics that summarize the topology and geometry any way that you want to. You want to use a statistical method that classifies each shape based on the summary stats. Again, any one that you want to. We want to derive an evidence of association metric. I'm going to show you this thing based on rate, but you can think about inclusion probabilities, the lasso, whatever your favorite method is. And then what you want to be able to do is once you find the significant topological or geometric summary stat features, you want to be able to map that and project those back onto your original object, right? So those are the 4 steps. Now, we've already covered 2 of them, right? I've always said that I'm going to use this Euler characteristic for this job. I'm going to stick in GP space because why not? I love GPs. And now we have to think about how to derive an association metric for these topological summary stats, okay? Now, the reason why my group focuses a lot on the like nonlinear models such as Gaussian processes, kernel regression methods, now like neural networks, is the idea that nonlinear methods kind of see the entire piece of like the pie, right? And the pie here is phenotypic variation. So, for linear models, we have, really nice hypothesis testing procedures, variable selection procedures. But the issue with it is that I have some kind of nonlinear variation going on. I'm going to miss that variation with linear methods that I'm going to see in nonlinear methods. The issue with nonlinear methods is I don't have the same variable selection infrastructure for free like I will get with linear models, right? So as a methodological person, what I think about a lot is how to build methods on the right-hand side that have the interpretability that we get on the left, right? And so one can think about this as the issue of the kernel trick, right? I have some high dimensional data. I can map that high dimensional data to some like n-dimensional function space. But the issue for a lot of us is once I've done my variable select, once I've done prediction in this like nice predictions on the right-hand side, if I want to understand how those predictions are summarized based on my input data, there's no like inverse mapping that takes me to my input space, right? So the idea of like the kernel trick issue is that once I use kernel regression methods, the classic idea of variable selection is lost, right? And I spent a lot of my time thinking about this in grad school, partly also with Young of, how do we build effect size analogs for this, for these approaches? And so going back to what I teach linear models and generalized linear models PhDs at Brown, let's go back to Stat 101. In a linear model, what do we have in terms of like an interpretability method? I have some regression model. I have an effect size. And effect size, right, is me taking my output and then projecting onto the column space of my data, right? You can think about this projection as being, generalized inverses, you can think about least squares, whatever the case might be. With those beta hats, what do? I go downstream and I think about how to do, I do hypothesis testing with them, right? And the effect size analog is quite similar, except instead of projecting y onto the column space of x, we're going to project our nonlinear function onto the column space of x. Okay, and that's going to give us what we call an effect size analog. And we can use the same type of projections that we do in linear models. But here, let's just stick to something so that these are equally comparable. Let's do to the same type of generalized inverse projection. Okay. Now, these beta tildes are things that we now can use as like weights for each feature in our data set. Okay. So I'm from Duke, so let's just be Bayesian about it, just because. So let's say that we set up some specify hierarchical model. You know, I can run MCMC where I'm estimating from a weight space view my Gaussian process model. Then I can also think about estimating other parameters in the data set. The important step here is that once I define a projection, I have some kind of deterministic step to kind of get an idea of sampling for my beta tildes, right? These beta. What I can do with these beta tildes now is I can think about things like posterior predictive distributions and things like that. And we show in one of our papers that these things are actually really predictive of if I were to think about how to sample out of test predictions with them. Now, this doesn't help us here though all the way, right? Because even though they're an effect size, it's hard to think about how to do variable selection with these beta tildes. They're just weights, right? So how do we think about ranking these weights in a way that is principled and makes a lot of sense? So those who remember how I think about this before during my last visit, I like to illustrate this with sports, right? And so those who are a little bit new, like I'll kind of walk you through how I visualize, and we use rate to do this in this case. We rank variables based on their relative importance from the other variables in the data set, right? And I like to describe this as looking at a team, right? So here, some of you might have recognized this if you watched The Last Dance. I have some kind of team together, collectively, this team, this has some amount of information with them, right? Now, let's say that I take number 30 and I put him on injury reserve, okay? Without number 30, my team might lose a little bit of its identity information involved, but in totality, maybe it's still pretty much intact, okay? Let's say I take another random player, like anybody, and I put him on injury reserve, right? Just a random person. And let's say he wants to go shoot Space Jam, or he's going to play baseball for a little bit. Without that person on my team, I might lose a lot more in terms of like my understanding of the network, yet without that person around, right? So relatively speaking, that person means quite a bit, right? Now we can quantify this via KL divergences, right? So here we can summarize the variant, the effective or the influence of every variant on the rest of them based on the KL divergence between the conditional distribution with that variable's effect being set to 0 and the marginal distribution with that variable's effect having being marginalized, right? And so the KL divergence is interpreted as being 0. That basically means variant J is not a key explanatory variable relative to the others, right? My information is still there. Alternatively, the KL divergence is 0 if and only if that conditional and that marginalized distributions are exactly the same, okay? Now, rate, or this relative centrality, is taking that idea with the KL divergence and putting it on a scale of 0 to 1. Why I kind of like this scale is because it gives us a natural null hypothesis. So rate's null, which is really important, is that every variable is contributing equally to the output, right? And the alternative is that some variables are contributing more, okay? The sports analogy here is that I have a bunch of bench players under the null, and the alternative is that I have a few superstars, okay? So let me kind of show you how this works in practice via a small simulation study. So here we have a data set. Let's just say we have 2,000 samples, 25 genetic markers. You can ignore the details. The key idea here is I'm going to choose 3 variables to be causal, okay? They're going to be my Jordan, Pippen, and Rodman, okay? What we can do now is run a GP. We can derive these effect size analogs, and I can derive rate from these effect size analogs, and we're going to rank our variables, right? So what the GP will say is, okay, I've identified Jordan, Pippen, and Rodman as being the most important players, okay? What's cool about this idea of rate is is this idea of centrality. So let me kind of demonstrate how this works with in terms of centrality. So I could take a variable, let's say I take Jordan. I condition on the idea that Jordan's out of the game And maybe then I go iteratively through every other player and say Jordan's out along with every other player iteratively. Okay. Now, when I remove Jordan and every other player iteratively, and we refit rate, what happens is the model still says that Pippin and Robin are my most important players. But if you look at it, the importance of every other variable has gone up, right? But it went up in like a uniform way. You can think about this like saying, I've lost 40 points. So those points have to come from someone. So the other players have to step up, right? Their importance, the relative importances all rise, but they all rise uniformly because they're just basically noise, right? We do the same idea where I take Jordan and Pippin, put them out of the game, and everyone else iteratively. I still identify Robin. You can see, again, I'm kind of hovering over this uniform 1/p line. Do this again, and I'm basically left with like this uniform distribution of just null players. Okay. Now I could do a sanity check and say, okay, what if I take a number 30 out of the game? What happens? Well, nothing. I just see everyone still around. And the idea then is, what if I generate things from just noise, right? You can kind of see everyone kind of hovers around this line. So we're going to use rate to rank our topological summary statistic features. And how this is actually going to manifest itself in terms of like surface shapes is actually kind of cool. So I'll kind of come back to this. But remember, the idea here is that under the null hypothesis rate, I have a bunch of bench players, and under the alternative, which means everyone's contributing equally, and under the alternative, I have some people that are contributing much more than others. Okay, and I'll show you how this plays a role when we think about shapes. So that's what we're going to use for our association metric. Now what we're going to do is we're going to project. We need to have some kind of projection. And this is actually probably a real novelty of this work, which is thinking about this projection machine. So our shape algorithm takes advantage of this idea that, you know, my goal is once I do variable selection to map it back onto my actual shape. The idea here that we're going to take advantage of is that when I'm rotating my object and I'm doing these Euler characteristic sweeps, directions within a given cone of each other probably have similar information in terms of what's significant, right? If I take a shape and I do a sweep and I rotate that shape a little bit to the left, I still also probably have similar features that were in this cone as being seen as being significant. And so we're going to take advantage of that here when we do our reconstruction, okay? So the reconstruction algorithm works as follows. I'm going to pick a cone of directions, some set of directions. I'm going to, for each direction, find all the vertices that correspond to the topological summary statistics that my GP found via rate. I'm going to repeat this for every cone. And then I'm going to take the union of these map vertices as my reconstructed map. Okay, so let me show you how this works as a visual. I have a shape. I have a direction of cones. I'll find the significant ones for that particular cone. I'll save those. I have another shape, or I'm sorry, I have the same shape, have another set of directions, find the significant features for that. And then what I'll do is I'll reconstruct my object by taking the union of these different cones as like what was significant in each cone separately. Okay, that kind of gives us more confidence that that a feature I saw for one direction that was seen in another direction is consistent across them. Okay, so that's going to be our projection. Okay, so that is how we're going to do our rate pipeline. Okay, now one hidden gem about this paper to me is doing simulations. Like simulations are hard. In genetics, we take that for granted, right? Like we have some genotypes, I can create some kind of synthetic phenotypes, I can create any kind of structure I really want, right? Like quite well. How do you take a shape and create an output that is connected to a certain feature where you know the proportion of variation that feature explains on three-dimensional surfaces is not as simple as it sounds, right? So we stick to case control objects because of that feature, and I'll show you how we have a few simulation frameworks that I personally feel like are like part of the true gems of this paper more so than even the method is. So the first proof of concept example here is we're going to take spheres, 100 spheres, we're going to break them up into two classes, 50-50. What I'm going to do is I'm going to take these spheres and I'm going to perturb them in certain ways. Okay, so I'm going to take shared cusp regions for all 100 spheres, and I'm going to, with some little Gaussian noise in these regions, basically derive some cusps. So those cusps will be shared across all shapes. Okay, then what I'm going to do is I'm going to have class-specific causal regions, which are going to be indentations into these regions. So I'm going to take red regions here in this region for this group of shapes, and another red region here for this group of shapes via some like random configuration via some noise, and those are going to create our causal features. Okay, and I can do this with like more indentations, more shared cusps, however I want to do it, right? And then what we're going to do is say, SINATRA, can you identify the vertices that are in the causal regions and not anywhere else in the shape, right? The same kind of idea of building a ROC curve like we would do in any area of like genetics and other areas of statistics. Okay. What's really cool here about this is, so here on the top, you'll see the different types of shape perturbations we do. We increase the number of causal and shared regions as we go up in scenarios. So scenario 1 is the easy case. There's only 2 indentation, 2 shared features, 1 causal feature. 2 is harder. We have more causal features. So you see little shapes get a lot more heterogeneous, right? 3, we have a lot more heterogeneity. You can see on the bottom, these are ROC curves for SINATRA and how it does for different parameter configurations. The idea here for the cones is that you want to do more rotations of your object to see more of it so they can have more power. But you can see the power of SINATRA dies as you move to more heterogeneous settings, right? As I move over to the left, the power starts to go down, right? The intuition here is much like we have in genetics. When I have one causal feature, there's a few vertices that contribute to the most to the variation. It's like a sparse architecture of my traits that we have in genetics. When I move over and I have more and more indentations, the effect sizes for each of these vertices get smaller and smaller and smaller, almost like a polygenic type thing, and it's harder to identify those features, right? And so there's like a really nice connection between this and what we do in other areas of science. So this is sensitivity analysis. What we had to do for method comparisons, because this is what AOAS wanted, was try to figure out methods that were similar. So we did some really like non-apples to apples comparisons here, which I tried to be very explicit about in this paper. We tried to find other methods that did something that was like kind of similar. And so on this very easy case, what you can see is like even like thinking about this maybe as taking straw man approaches, which is like I could take the XYZ coordinates of a shape and maybe do lasso on the XYZ coordinates as each individual feature and say like, okay, let's just take the mean or the max of these effect sizes, and then those will be my features that I rank, right? And so you can see that these very easy approaches also do quite well for this very simple shape analysis, okay? Now let's say, okay, let's make these simulations a little harder, right? In genetics, you would never just say, I'm gonna take random genotypes and then walk away. It's like, let's have some real structure to this, right? So how do I do that? Well, we got kind of interested. Oh, so let me, before I get to there, we did one more experiment here, which is do a null hypothesis. So this goes back to my rate example. Rate, remember I said, the null hypothesis is that everything contributes equally. The alternative is some things are contributing more, right? So what does that mean? Well, in a shape, there are two null scenarios. There's the first scenario, which is every shape looks identical, right? Almost by like, there's nothing really different here, maybe like a little bit of variation, right? So rate is going to say everything looks exactly the same. And what SINATRA is going to say when it reconstructs things is like, well, everything's the same here. If you're looking for enrichment about like what's different, like they're all about contributing equally to the difference. And so everything looks exactly the same. That's null hypothesis number 1. Null hypothesis number 2 is the alternative, where I have a shape that's of one thing and then I have a totally different shape of the other thing, right? They look completely different. Well, SINATRA is going to say, well, everything's equally contributing because everything matters here. And so everything should be reconstructed exactly the same. And so you see the same kind of null case scenario, right? So what I say about SINATRA in terms of its contribution is its space is really in between those two scenarios. If they're exactly the same, like, I don't know why you're doing this in the first place. You probably maybe should do something else with this shape. And if they look totally different from each other, you don't need SINATRA to see the difference. You can see the difference, right? So SINATRA kind of lives in the middle where there might be structural variational things that are going on, but they're not necessarily totally seeable by eye, but also you know that something is there, right? So let's do a simulation that's a little bit harder, okay? So this is a characterization scenario, and I won't stress you with this, but effectively what we do is we take real teeth and I'm going to make caricatures out of them, right? So here's a real tooth. What I'm going to do on this tooth is I'm going to find landmarks on this tooth, and I'm basically going to elongate them or make characterizations of them. So for group 1, I have a certain meta landmarks, and I basically blow them up in some way. That'll be shape class 1. I'm going to take another set of landmarks from the same derived tooth from shape class 2, blow them up in a different way. And I'm going to ask SINATRA in the third step, reconstruct this shape and find the vertices that matter. As you can see how well it's kind of doing. this is more of a realistic case where I have like almost like a phylogenetic relationship from these teeth, right? And I'm asking you to find the realistic differences between them from like almost like an evolutionary process. So same kind of idea holds from this first scenario. When I do this with fewer landmarks, I have more power. When I do this with more landmarks and more heterogeneity, smaller effect size, I have less power. So the same kind of thing holds. But the interesting thing is when I do these, the strawman approaches now no longer work, right? So actually the lasso idea with the vertices, like they're almost like completely random now. They're just strictly on this line. You can't just take this vertex, do like a lasso based approach here. The limit shapes algorithm is from this group at Harvard, which is not made for this, but it works quite well only when it has correspondence maps. So if I tell the limit shape algorithm, this vertex corresponds to this vertex and it's able to know that, it does quite well. The second you jumble that map up by 10% or a little bit more, it completely drops off in terms of power. And so that decrease you're seeing in the blue line is when you jumbled up its understanding of these correspondences. SINATRA doesn't actually need correspondences to work because it works in topological statistics. And so for the last couple of minutes, I just want to talk about this idea of recovering known variation and one really cool future thing that people might be interested in. So here we take morphological variation across these different primates. I have 59 molars from these different types of primates. This paper was hard because people haven't thought about this question a lot. So what we did is we tried to do like a pseudo ground truth example where we took a tooth from the tarsius where I know based on evolution that a certain cusp in its lower molar was preserved because that's how it eats and digests food. And the other primates don't have that anymore, right? So what we did was we compared whether SINATRA was able to identify that lower cusp as like our ground truth feature, okay? And then we looked at these features kind of differ across. And so these are these look phylogenetically. That red dot on the red region on that bottom for that tooth is the cusp we're going to kind of surround our analyses on. And then what we did downstream is say, okay, how well does SINATRA identify that? And then we did these region of analysis, of interest analyses where we say how often was SINATRA finding that by chance, right? Like what's the probability you're identifying that by chance? Now, a real hidden gem in this process came to me from a reviewer, which really challenged me, which is like, you claim SINATRA can do this with no correspondence maps, but you're pre-aligning your data set. So like you kind of have correspondence maps, like implicitly. So what happens if you did this non-aligned shapes? And this to me is something I really want to highlight. So if you see me raving about this part on Twitter when it comes out, because I think this is part of the coolest part about this project, SINATRA actually works, you could actually align three-dimensional shapes in topological summary statistics space implicitly. So what you can do is I can take an unaligned mesh, unaligned objects, a bunch of them. I can run Euler characteristics on them. So I have a bunch of curves from unaligned shapes. If I minimize the distance and normalize these curves, that's like normal, and then I reconstruct the objects, I'm actually normalizing the shapes themselves. And so what you're seeing on the top of these unaligned images, I've gone through and then I align their topological summary system instead, and I reconstruct the meshes, and this is what they look like. It's not perfect. We didn't perfect the algorithm. We just tried to show people that you could do this, but this is kind of what you get. And on a grand scheme of things, this is what the aligned mesh data set looks like. It's actually not that bad. And so future work for us is thinking about like, how do we actually perfect this quite a bit? And so we're working and writing a paper now and thinking about alignment via topological summary stats. You can think about this being very cool for big high dimensional data, where maybe I have multi-centers and I want to send this somewhere and send my data somewhere, but maybe that data is too large to send. So maybe you just send the topological summary statistics instead. We can reconstruct this in a centralized location and think about alignment and stuff without having to think about confounding bias that may come from these different places, more so than what already will come from like with the different image machine. And then you can see how well SINATRA does here. So I just want to finish off with like future and ongoing work, because I just think it's cool. And if people want to think about this hard stuff with me, I'm all about it. So, you know, eventually what I want to do is get back to radiomics and this, where my motivation for this came from. And so I still want to probe this idea of if wind shape variation is correlated with genotypic or phenotypic variation. The issue with this is a hard problem. And it's hard because in the teeth, we actually have this really nice setup where phylogenetics has done such a good job. Like genetics and selection does such a good job where if I'm a monkey that eats bugs or plants, my teeth are going to be shaped differently, because if I don't have that morphology, I'm not going to be able to digest my food and I'm not going to survive anymore. And so intra-class heterogeneity is low. When you move to tumors, that's not the case. They can grow however they want. And so the intra-class heterogeneity across these within a given tumor class is quite high. And so when you think about how to do SINATRA, you have to think about how to maybe deal with these types of variations that you didn't necessarily have in this first case. So that's something we're really trying to think about now. And I have a way to think about this a little bit, but I'll say that for the very end. And the second thing about this is extending this topology ideas to more imaging and maybe not necessarily shapes. So we have this idea of treating images as fields and doing the same kind of topological summary things on things like PET or 3D microscopy and things like that. So this is other kind of areas that we're pushing on now. And developing topological invariants in order to do these kind of things. And so, one way I've been thinking about this is I kind of move to structural biology a little bit because, why not? But why not add one more thing to stuff that I like to do? Proteins are very cool because they kind of sit in the space of very low interclass heterogeneity or rigid structures, or depending on the mutation, very flexible and very heterogeneous types of structures. And so we've created this thing called SINATRA Pro, which does almost the exact same thing of identifying structures that are structural fingerprints, biophysical signatures that are attached to different mutation in proteins. And we've been doing this over a wide range of them and actually recovered a lot of really cool biology type stuff. So SINATRA Pro is like our first attempt as we kind of inch our way back to medical imaging. And so we've been thinking about this idea of biophysical signatures and protein dynamics. And so that's been like super fun. So with that, I know I've been talking for a super long time. This is like a huge collaborative effort. Like I've had a lot of fun working with people who are like, this is really hard. Let's just think about this. And so this spans across a lot of people in different places, from Brown to back at Duke to Microsoft Research and other places. And people have been nice enough to think that this work is someone interesting and give us money, so I'm appreciative of that as well. So with that, here are some references, and yeah, I'm happy to take any questions or comments people might have.
Thanks. That's great. If there's any questions.
I had a question. So the really nice property of the Euler characteristic transform is that you have that invertibility. But that is, that theorem holds, when you have a smooth manifold. And, what we're doing here is we're approximating that smooth manifold, starting with the sublevel set filtration, So I can imagine some ways where maybe that could become problematic. So you're you're choosing a granularity of those that sublevel set filtration, right? And then also you have a fixed number of sweep outs that you're doing to then extend that to 3 dimensions. So have you looked into, whether you're getting a sufficiently high resolution approximation of that smooth manifold to where you
Absolutely. Yeah, that's the thing that we in every paper that I've done on these topics, we talk about the pros and cons of these discretizations via the sublevel sets, and this idea of that parameter choice is going to be very variable based on the shape that you're looking at, right? And so in the top paper, what we do is we actually do a nice sensitivity analysis, and I actually made a table which I thought the reviewers had a really good suggestion about, of trends of how to pick these parameters for certain surface and manifold types. So you might need more directions for certain things. Going back to my teeth example versus the tumor example, you want to be careful of the distance between your sublevel sets, right? You don't want to, if you're looking at the teeth, you have more global features. And so being too fine in that filtration, you're going to lose signal. You will have washed that away, versus in a tumor case, you don't want to be too granular in that because you might skip over very local things. And so, yeah, there's a So there's a lot of work that we are doing to thinking about how to pare down what you might need in practice in order to like not be so dependent on some of these like free parameters that you're going to have to choose, where this could be an issue. Yeah.
Cool. Yeah, I guess that's the thing with using the sublevel set filtration. Like you get this nice, Euler characteristic that can be used. But I'm guessing you thought about like if you use like a check or Vietoris-Rips filtration, where you can then, compute persistent homology directly on your space without the sweep outs. And like, as long as your persistence, your like max radius is half the greatest distance between 2 points, you get like the full picture.
Right.
So that might not be
the idea that I think where everyone wants to move, or what I've been pushing for people to think about when you think about this, is ideally you would do the persistent homology transform on stuff. Like if you actually had a nice way to do the invertibility question, which people like Brittany Fossey at Montana, other people are thinking about. I think what you're seeing from those groups is like it's hard, and so like, but that would be the thing that we would ideally like to do if it was available to us, for sure.
Yeah. And I think then you can look at, like you're mentioning low persistence. I think then like low persistence captures curvature in some ways. But yeah, it hasn't been people haven't quantified it yet in nice ways.
Yeah, but that future research is something that's that I've been pushing for people to also think about too, because it would absolutely benefit us as well. Yeah.
I have a question. First, thanks for the very awesome talk. I feel that I learned a great deal today. So I have 2 questions. The first one is about the choice of number of directions. I know, directions that have a small angle, likely they'll produce similar blocks of columns in your regression setup. How do you usually choose the number of directions?
So there's a recent paper, I think that's still on archive, and maybe in its last revision at one of these mathematical journals, where they actually compute a lower bound for a given surface and a number of critical points on your shape, how many rotations you should do. They have like a lower bound for that number. What we typically do, as you can see from the empirical Sprott curves that we did, you want to be more in practice. You want to do you want to be liberal in that choice. You want to choose more, even if you don't think you'll need that many, because the power that you'll get will like asymptote at some point. So there'll be some amount that you can do. You just can't do better than that, because the Euler, like we were just talking about before, the Euler characteristic is going to be a summary stat. In some cases, it'll be sufficient in some things, but it may not. It's going to give you everything that you can possibly get, and so you'll peel off in terms of your power. And so what we say to people who are running this, and we try to highlight this everywhere, is choose more directions. Choosing one is not, you'll have nothing. You won't get any power, and so you want to choose more, as many as possible. I think in the tumors, we only did like 70 or something. We kind of showed like in a sensitivity analysis, like 70 was fine. And then in the shapes for the teeth, I think we also did something similar to that. And we did a full blowout simulation type thing to kind of show where you would lose a lot of power. Directions are easy because you just choose more. Like that's the rule of thumb, is just choose as many as you can, right? Filtrations and stuff are the harder ones. Those are the ones where you have to think about your actual domain and science thing.
Yeah, thanks. Another question is about the comments you had, the comment raised by the reviewer you mentioned. So it's about comparing the limit approach and your approach.
Right.
So my understanding is that you always do the spatial alignment first.
Right. Yeah, I love talking about this.
The reason why I asked that question is, I wonder, after alignment, you do not extract any correspondence. You just do the sweeps, and then that beats limit, even if.
Right. So the algorithms that we had were not doable without the pre-alignment first. Yep. And so we did all the alignments first, and then we showed if you didn't do alignments, where you would see power for stuff. And then we showed the correspondence maps for the limit shapes algorithm, actually, we didn't have them. So we had to construct them ourselves, which took forever. And then we were able to run the limit shapes algorithm. You could imagine if you didn't construct those correspondence maps correctly, which is what we try to play out a little bit in the simulations. I also want to mention, this is like recording and I want to be fair to them, like this is not what the algorithm was built for, it's built for something else. A reviewer said it might be good to maybe think about something like this. So we took their algorithm and worked with the authors to try to make it as apples to apples as possible. But we also just showed, like, if I didn't have that correspondence map correctly, what might happen. Then what the reviewers really pushed us on was in the real data, like show in real data that you can get results that are similar to when you similar for SINATRA where you compare it across these different teeth. And so then we explored, okay, how you might align the shapes a priori before running this thing in topological summary statistics space, and then going from there. And that actually has a really cool application even down the line. So I have a student now who's thinking about this from a protein alignment question and these other kind of things. And we're really trying to think about how to really make that algorithm a lot better. What we do here is pretty, I don't know, it's not as sophisticated or as maybe nice as one might like it. It's a pretty crude algorithm, but I think there's space for future work there for sure.
Thanks.
Yeah.
That's a good question.
Yeah.
I think this might be sort of a naive question in topology land, but am I right in thinking that the Euler characteristic is insensitive to the gross scale of the object? That if you take the same object and blow it up, it's got the same Euler?
Right. So there's a really good question of when you move to certain areas where you would want to also know geometric information. Yeah, that's a great question. So there are two areas where SINATRA on its own probably will be insufficient and probably not ideal. The first area I think is radiomics on its own, where you probably want to have idea of size, because size tells you something about aggressiveness of a tumor sometimes. And then also things like PET and microscopy and stuff, things where you have texture, where texture also might mean something. And so how do you kind of build, instead of, like, I remember a reviewer in the JASA paper for the glioblastoma thing said, why are you separating these data types? Wouldn't it be ideal to combine them? And I think moving forward, that's something someone would probably want to do. I've thought about interesting ways of, like, maybe what you want to do for tumors is, like, you condition on geometric properties and then you run topology as, like, the features you want to actually learn from, so that you can kind of do variable selection that way. Yeah, there are a lot of interesting places there, but I think topology on its own and some of these other domains that we're trying to move towards that are a lot more sophisticated, it's not going to be sufficient on its own.
Right. I mean, it seems like in some circumstances, something like size or texture is important and you really want to know it, right? Like tumors, a bigger tumor is different, but when you go to, like, the teeth or, like, heel bones or something, you want to capture something about the structure and not how large the animal is.
Right.
Or I think brains have the same issue, right? Like bigger people have bigger brains, but
yeah, so that's a interesting domain thing that we were also kind of thinking about. Proteins were also kind of absolved from that. So proteins are nice because we include this kind of molecular dynamic situation. So kind of have this, like, short-term evolution thing where a lot of things can maybe happen over a short-term scale. You could think about how that might project onto things like a tumor or something like that. But you also don't have the same kind of issues that one might have with the tumor. So we don't have to worry about size of the proteins and these kind of things. Like, we're just focused on purely structural biology in that sense. So I've been using it as an intermediate play as we move along, because we're like kind of adding incremental, like, challenges at a time to kind of be able to ground ourselves. The one thing I'm also really interested in that we haven't been able to do with the tumors that I kind of briefly touched on earlier is generating shapes with a continuous output. is something that I think is like, I know it seems people may not appreciate it. It's like, it's just like a simulation kind of thing, but like building likelihood-based models for shapes is something that I think will really help us be able to assess these kind of methods in more principled ways. Where right now we're just limited to case control categorical things because it's easy to think about how to divide variation there. But then, this idea of, for people, going back to the glioblastoma, I have a shape, and then I also have an output that's continuous in terms of like days free survival or drug efficacy or something like that. And so how do I think about how to model something where I'm controlling a continuous output with like a physical feature where I'm kind of controlling that variation like we do in like something like a GWAS or something with our simulations, right? Yeah, and that's hard, but I think that's something else that is like a pretty cool thing that I hope people think about.
Yeah, I could imagine with the simulation issue that there would be like a clear temptation to just simulate from your model, right? Where you say like, okay, I know the Euler characteristic of this object and I'll just simulate my feature from that, but you've sort of cheated or something in some way when you do that. So
yeah, we wanted to be we wanted to do that. Like, I think that was like our last resort. And we spent a lot of time thinking about how to do this and like how much noise to apply. And we started with like Brownian motion at some point, and we kind of like moved on. Yeah, it was, I know it's something that like, I think we kind of mentioned in the abstract now that like this is something that we're actually kind of really proud of. But even there, like, even having more systematic ways of checking some of these approaches, I think would be really good, that I don't think we've spent a lot of time on previously. But even then, I think this would be pretty cool to as a future direction. Yeah. Should I stop sharing my screen?
One more question for you, but I want to give space for other people if they have questions. Okay, barring other questions from other people. So I'm wondering does in principle the same approach work in more than 3 dimensions? I'm thinking about time maybe as your 4th dimension. Could you just sort of naively say it's 4 dimensions and I'm going to run it through the same, through a analogous pipeline? Or
yeah, so the proteins we do through time. So we have 2 proteins that we start with, wild type and a mutant. I guess you could do mutant. We have these MD simulations where we run these mutants over some period of time. You collect these structures, and you could do SINATRA to see how basically get these like biophysical properties of these like short-term evolutionary tracks to kind of see like how structural differences happen for protein A versus protein type B. It's easy to think about for 2D, for 3D. For 2D, I think it's a little harder. Yeah, the 2D one has been tough, and I think we have a way of doing that where instead of treating a shape as a shape, you treat an image as a field, and you do this over the field instead. I think that's been the most promising way that we've thought about this, and I think we have some preliminary results on how this might work. But things like microscopy up to this point were challenging. If you just treat the microscopy images as like a shape, like I don't think that's the right way to go. I think you need to treat 2D as a different type of data and then run it that way. I think that's probably a more, yeah.
That makes sense.
Yeah. So yeah, I don't know. It's like really fun stuff. The thing I love about the TDA community is like super small. So there are people that like you get to know everybody quite well, but also it's like a super collaborative community where people are like always open to try like really hard things. And so it's been fun to work on this stuff.
I think I missed what the acronym TDA is for.
Oh, topological data analysis. Sorry. Yeah,
okay.
Yeah. That community is an interesting one. They have a community-wide Slack, which I'm on, and people just go on there and share ideas. It's like it's a weird thing. It's like all year round, like people go on and share ideas, share their papers. So like I had this idea, do people want to work on this with me? Like it's like night and day compared to how like genetics would operate, right? Like where people don't want to talk about anything you have until it's basically out. It's a really interesting group of people, but I actually love it. It's fun to like maybe not be so protective of how you think about science for a change. So
that's great. Yeah, that would be fun to have in other fields.
I know. Yeah. As I tell you what I liked about ProbGen is kind of this, like, at least for like the week, people kind of let their guards down and just talk about stuff. And I think that's like super fun. It's kind of like this understanding that like some people are going to share some things that are not out yet or whatever. So let's just like talk about it. Yeah, it'd be really great if more fields like kind of took on that kind of identity and role. So yeah, hopefully maybe one day in the future.
So were you at Duke and like just had a conversation with like John Harrar or something? Like what was like the first time?
Oh yeah, that's a really good question.
There's a lot of people there doing stuff, but
yeah, Ingrid and other people. Yeah, I had known them for a while, and actually, was it John? Someone was giving a talk at Princeton, and my advisor was going, and he was like, "Hey, do you want to come along? I think you would find this stuff kind of cool. I don't know, just it was like the shape of cancer, I think is what it was called." And so there were people coming in who were wet biologists, and people coming in who were like pure mathematicians, and they were talking about like where they can maybe meet in the middle. Looking back on it, it was a little extreme to think like a pure mathematician and someone who's like a cell molecular biologist were going to find somewhere to meet in the middle, but that was the goal of like that you could do like IAS, right? And so I went and I met this man, I met Raul Rabadon, whose group is like a blue physicist at Columbia. And I remember he did a talk and I was like, oh, this is cool. He was doing stuff on resistance. And I was also doing kernel methods for therapeutics and resistance. So I was like, oh, it'd be so cool if I just came, or if I could like, talk to you more, maybe Skype when I get back to Durham. And he's like, why don't you just come to New York? So my advisor was like, yeah, I'll fly you out to New York. So he just like, I got on a plane, I flew out to New York. This woman, Anthea, who was a visiting professor with me, was going, she had gotten a position at Columbia. So she was like, oh, I'm going to find out more about the university. So we went, she took a job there, and it was like, Raul was like, come back and just hang out. So I would fly up to his lab like 10 days at a time and like just kind of learn TDA from like these physicists and like people that were like doing this really cool stuff in cancer. And then we wrote that Euler characteristic paper together for glioblastoma, and it was like a lot of fun. And so then I just started asking these other questions, and yeah, he was great. He was in my third and fourth year, I was just like up in New York like quite a bit, kind of learning from whatever it was that he was doing. And so
that's crazy though, because like there's so many people who come out of Duke doing TDA,
yeah,
but it's like you were like doing it from Columbia.
Yeah. It's funny because like Duke Stats is here and like math is here, but like I don't know, you can never see anyone walk in or out of that building. Like the math people are kind of just like in their own world. And now I know a lot of them like quite well, but like when I was there, it was like you never saw them. Yeah, and like hindsight is like, I don't know why I'd never like just went into my own backyard and was like, hey, can you talk to me about some of this stuff?
Yeah, I do agree though. So like, I came from a department of mathematics, and I've been working with an algebraic topologist on some TDA stuff, but it was me, an algebraic topologist, and a neuroscientist. And so like just figuring out like going between the pure math and like our application fMRI was like, I mean, it's pretty cool though.
Yeah.
Yeah, we have some stuff around here mentioning like using the precision homology transform.
Mm-hmm.
And yeah, so like when you do like Vietoris-Rips filtration, yeah, like that mapping back is unstable. Like whatever cycle representative you have might not be, it's numerically unstable. So yeah, we were working on that problem, and we have something where you actually, instead of looking at one cycle representative, you look at a larger equivalence class of them, and we think we have maybe something useful from that.
Very cool.
But it's hard, man. It's hard, and it's very computationally expensive.
Oh, really? Okay, yeah. I've always imagined it would. I imagine the first version of this would be, but I still think whoever is able to like give some viable solution, I think it'll be super impactful for the field. That'd be cool. Yeah, if you have a paper on that, I'd love to read it.
Yeah, I have. So Yeah, I have a sketch of what we're going to write as a paper as an abstract that was at a conference a couple months ago. Yeah, I can send that over, just like the general idea of it.
Yeah, that'd be awesome.
Also, yeah, when I email, if you can add me to that Slack, that'd be cool.
Oh yeah. I've been like, I can't believe people don't know about this. Like, oh, this is like it's like underground community. So someone added me, and like all my friends are on. It's like, oh my gosh, it's like a whole like thing of people. Yeah, I'd be more than happy to. And people like share jobs and all kinds of stuff. Fellowships, like if you ever like want to go like to Max Planck or something for some stuff, like people are like, come and work with me on things. It's unique. It makes me think like how progressive other fields could do if we adopted like some of those ways of doing things. So
it helps that they're small though, like, you know, helps.
Yeah, that's true. That's true.
Yeah,
that's true.
Hassan, I have to make a call before 5. I don't want to end your conversation though. If I can make you the host, I'm gonna do that.
You know, I think I'm ready to.
Yeah, we'll talk offline.
Any longer either.
Gene, thanks so much for inviting me. It was really fun.
Yeah, I'm really glad you came. When I knew I was organizing this seminar, you were like the first person. I was like, I'm gonna invite Lorin.
No, thanks. I really appreciate it. Hope it
All
was
right.
fun.
Take care,
yeah,
everybody.
actually, put me host for like 1 second.
Okay, all right.
All right, let me just do you mind if I get your email?
Oh yeah. Can I put in the chat? Is that easier?
Yeah.
Yeah. This is probably the easiest one. Oh, this went to. Sorry, this switched over to.
Cool. Okay, so you want the Microsoft in there?
Yeah.
So I know sometimes people have multiple and you don't know which one.
Yeah, I do have multiple, but the Microsoft one is much better at alerting me that I get emails. It like comes directly to you and it like makes a ton of sounds, and it's just like, okay.
Yeah, cool. Yeah, no, this was an awesome talk. I think that like I'm always seeing people either more in math or like, you know, really heavy application, but I feel like, you know, you're like kind of in between, this nice in between, and there's like so much good that can happen there.
Yeah. Well, let me know.